RNA splicing starts with pre-mRNA, which already has the 5' on it, which creates stability. snRNPs and other proteins will then join together to create a spliceosome. The snRNPs contain snRNA, which will bind to various nucleotide sequences on the intron, causing the snRNPs to act as ribozymes, which will cleave the intron out of the pre-mRNA, and the intron is then degraded. The remaining exons are then spliced together with the help of other proteins, and the addition of a poly A tail creates more stability. It is then necessary for the mature mRNA to exit through the nucleopores. This diagram represents the regulated synthesis of repressible enzymes in the TRP operon in E. coli. This TRP operon is usually on, which means that the repressor protein is by default inactive, which means that it cannot bind to the operator, which means that RNA polymerase can bind to the operator for RNA transcription to occur. When RNA transcription occurs, it creates polypeptide subunits that will code for enzymes that eventually make tryptophan. When tryptophan is accumulated in excess, it acts as a co-repressor and will bind to the repressor protein, activating it and changing its shape so that it can now bind to the operator, preventing RNA polymerase from making RNA. This means that no transcription occurs and no polypeptide subunits are created. TRPR is the gene that's located upstream from the rest of the genes, and it codes for the repressor proteins in the TRP operon. This diagram represents the regulated synthesis of inducible enzymes of the lac operon in E. coli. The lac operon is usually off, which means that the repressor by default is active, which means it binds to the operator, preventing RNA polymerase from making mRNA when lactose is absent. However, when lactose is present, it is inactivated by allolactose, which binds to the repressor protein and acts as an inducer, changing its shape, and so that it is no longer able to bind to the operator, leaving the pathway open for RNA polymerase to pass through and transcribe genes to make mRNA. Now, LAC-Z codes for beta-galactosidase, which is responsible for converting lactose into galactose and glucose. LAC-Y is responsible for permease. Permease is a surface protein which allows lactose into the cell. LAC-A codes for transacetylase, which aids in the catabolization of lactose. Now, LAC-I is a gene that is upstream from the rest of the genes, and it codes for the repressor. Activator proteins bind to the distal control elements on the DNA, and the distal control elements are grouped together as the enhancer. The 3 activation sites make up the distal control elements. The DNA binding protein binds to the DNA and causes the DNA to bend over so that the enhancer is close to the promoter. Mediator proteins, general transcription factors, and RNA polymerase II are all nearby at this point. The mediator proteins will bind to the activators, and in turn, the general transcription factors will bind to the mediator proteins. And in turn, RNA polymerase II will bind to the general transcription factors. This ensures that it is positioned correctly over the promoter. This is now an active transcription initiation complex. and it allows for RNA synthesis to be amplified.